Okay, so we can get started. Hi, everyone. Today we have an exciting topic. We'll be talking about the response to adoptive cell therapy using tumor-infiltrating lymphocytes and why some melanoma patients responded while others didn't. We'll analyze the clonotypes from baseline tumors before and after treatment using a single-cell RNA and TCR sequencing. And then we'll be discussing the results and what separates responders from non-responders. Anyone who is studying immunology or cancer research is absolutely going to learn something today, as we will even be going beyond the study and giving you practical tools to enhance your own research. Let me introduce today's speakers. First, we'll have Sandra Woods, CEO of ImmuneWatch, talking about the paper and key results, and how to go deeper. And then Sam Puzlowski, the CEO of My Laboratories and creator of MyXCR, will replicate the study and show us new findings. If you have any questions throughout today's session, feel free to drop them in the chat, and then we'll dedicate some time at the end to answer those questions. So Sander, I will now pass it off over to you.
Okay, let's get this started. You should be able to see my slides now.
Yes.
Great. Well, thank you so much for this introduction, Monica. Today, we'll be talking about TIL therapies. TIL stands for tumor-infiltrating lymphocytes. And even though the concept of TIL therapies is pretty simple or very simple, these are an innovative and very promising form of immunotherapy. So basically, you start by taking a tumor biopsy from the patient. You then isolate the tumor-infiltrating T-cells or the TILs from this biopsy, then do in vitro expansion in the lab, and then reinfuse them in the patient again as a therapy. So it's a pretty simple concept, but a promising therapy. And today we'll be diving into one specific paper called Tumor-Reactive T-cell clonotype dynamics underlying clinical response to TIL therapy in melanoma. This paper comes from the Kukos lab in Lausanne, Switzerland, and actually focuses, of course, as the name says, on TIL therapy to combat melanoma or skin cancer. And the main question of the paper can be put in very, very simple terms, maybe not so scientific, but sometimes TIL therapy works, sometimes it doesn't. Why is this, or why is that? And so using the graphical abstract, we can take a quick look at the main results. So here you can see in the upper left corner a cartoon for non-responders. And on the bottom right, you can see a cartoon for the responders tested in the clinical study. And each section of the tumor has different T cells sitting around. So researchers take small biopsies, isolate these T cells, and then expand them in the lab using isolation and then expansion to create a cell therapy product. And what they found is that for the non-responders, the final cell product has many blood-borne CD4 T cells depicted here in green, and non-tumor reactive cells here shown in blue. For responders, on the other hand, the product contains many more tumor reactive T cells and also some non-tumor reactive cells in blue. So when these products are infused back into the patients, responders see tumor shrinkage because they receive many tumor reactive T cells, and non-responders do not, and the tumor might progress actually further. So in a nutshell, this is just a graphical abstract, you can see at the tumor level, non-responders have few tumor reactive CD8 T cells, while responders have many of them. In the cell product level, non-responders see an enrichment of blood-derived CD4 T cells, while responders see an expansion of tumor-resident and tumor-reactive CD8 T cells. But after therapy, so after the cell therapy, biopsies from responders show high levels of tumor-reactive CD8 T cells, while non-responders show few. So I have to admit, pretty cool paper, right? So what can Platforma and ImmuneWatch actually add to all of this? So first of all, I would say using Platforma, you can actually replicate such a study easily without having to touch the command line. So perfect if you want to take analysis into your own hands. But then as a next step, using ImmunoDetect, which is also integrated in Platforma, you can answer one quick question that was also alluded at in the discussion, meaning what specific antigens are recognized by these tumor-infiltrating lymphocytes, by these TIL. And so as mentioned here, the specific antigens remain to date elusive. So today we'll take a quick peek into what such an analysis might look like. And for this, I will give the floor to my laboratories to Stan.
Thank you very much, Sander. Thank you for great introduction. Let me share my screen. I sent request to share. Again, let me share. Yeah, and what I will do right now, I will just start like in an interactive hands-on way, start the analysis of the data presented in this publication, and together during this course of the analysis, we'll try to get some of the insights and reproduce some of the insights and some of the results found in the study, and B, as Sander proposed, enrich these results with the detect tool and give and then get more. So Dimitri, if you can mute yourself.
Oh, sorry.
Muchas gracias. Okay. So, well, here on my screen, you see already some pre-processed data with Platforma. The first of all, what is Platforma? Platforma is a software which has an interactive graphical user interface, obviously, where you can get really deep insights from the data, like we see here. This is actually a space of all T-cells from all the patients from this study. And as you see, when I highlight some of the T cells here on this clonal space are highlighted, and the ones that are highlighted are actually the ones that are enriched with the therapy. So those are that were enriched in the product. And as a tooltip, you see the predicted antigen by detect tool. Yeah. And we can zoom in and we can see some nice things here. But this is just, you know, as a preview of what we are going to get at the end of this analysis. And now let me start from the very beginning and just go step by step in the way how you do this analysis and get the results. So let's first create a project. So webinar online. We start our analysis, our journey with samples. That's the first thing that we are importing into the project. And I have my samples, my FASTQ files located right on my laptop, but you can run Platforma on your server, on your HPC cluster in your institution or organization. And then, of course, it will run the heavier part of the analysis on that remote machine. So I'm adding all of the samples from the study. I'm importing them into the project. And then as you see, the samples and data, that's the first app, the first application, the first block on the project, automatically infers the sample structure, the sample name from the file name. Yeah, I'm going to modify this pattern to extract only this first part of the sample as a sample name, and also to match only the yeah, well, basically that's it. And only to match the TCR beta samples that we have here. So let's call this data set bulk. This data has also single cell part of the data, and we have capacity in Platforma to process this data as well. But this is a bulk samples, bulk TCR beta samples. And that's it. We have our samples, our FASTQ files. And we can also import the metadata associated with the study. And this metadata is actually what is very important to get the answers from the study here. So I'm just bringing the Excel file, a TSV or CSV file with all of the properties about our samples. And in this specific study, we have the patient, we have the time point, day 0, the CDP, and day 30 after the therapy. The material, and also the marker of the patient, partially responder, non-responder, and there are a few patients who are complete responders. So with this, I have the setup of my project. I have all of the data, and this is the very first step we are doing. The very second step, basically, in the analysis is running some QC metrics, for which we have FastQC application. But since this is T-cell data, we have MyCR app, MyCR clonotyping block, which actually does the QC internally. So I'm not going to run FastQC here. We select our data set. We select the library preparation kit, which was used for the analysis of this data. It's Oncomine RNA TCR beta short read assay from Thermo Fisher. Yeah, we know for sure that only TCR beta was sequenced here, so then we can restrict the number of receptors and analyze all the TCR beta. And there are some advanced settings. The one which I always recommend for everyone is for dry run purposes, just to test, to play with the data before running on a full scale, which may take some time, like depending on your infrastructure. I always recommend to start with, let's say, 100,000 of reads from every sample, just to get a sense what is there in the data, like a dry run. And then, of course, you remove this on the full run and run the whole analysis. Yeah, and run. If you configure Platforma to work on your server or cloud or HPC cluster, the analysis of all samples, and here we have 65 samples from, I believe, 10 or 12 patients, then the analysis will go in parallel. For every MyXCR run, the backend, the Platforma will run a dedicated instance for computing, dedicated job. And this app, MyXCR Clonotyping, provides a nice overview of, well, first of all, the quality of our data. I mentioned the quality control. So here we see some alerts, some really bad samples. Yeah, in this sample, for example, if we go here, we see that the majority of reads were not aligned. There are no hits. So meaning some problems either in the wet lab, or it depends. And then you can either exclude this sample from the analysis, which should be the case in case of all when we have this, or go back to the wet lab, resequence it, and do something. Yeah. So so this is MyXCR clonotyping. It generates some nice reports, as I showed here, quality checks. You can go deeper into the logs of the run, to some more advanced reports, etc., etc. But once the analysis is complete, and it's now complete, we can, what we want to do, we want to look on our repertoire, on our clonotypes. And for that, we have a clonotype browser app. I'm using the development version of clonotype browser. It has some nice features which will be published towards the end of this week. So this is like the basic thing, the basic block we're going to use just to look on our clonotypes. Yeah, so we have samples, and for every sample we have a table of clonotypes which we can download once it's ready, or we can go to the overlap where we see the whole set of all T-cell clonotypes that we have in the data. In case of my analysis, I used the sampled data. It's 874,000 of clonotypes. Yeah, and many of the clonotypes, obviously, since this is a time tracking project, we have patients and we collect data from different rounds. Yeah, many of the clonotypes actually present in many of the samples. There are some clonotypes which present like in 10 samples, etc., etc. So this is a big, boring table. How else we can look on our space of clones? A more fancy way to do this is to use clonotype space. This is a nice app, a nice block we have to have a bird's eye view, like a visual one, on the space of all our clonotypes. I'm running it. It actually should take some time on that scale of data, 800,000 of clonotypes. But since I already pre-calculated this in a separate project, it's faster. And this is actually a UMAP where 2 points, each dot here represent an individual clonotype. And the idea of this space of this UMAP is that 2 clonotypes located here on the plot closer to each other if they share common properties. And those common properties are defined by their protein sequence. So this is already something. We see some structure. We see some structures. We can actually play around with the parameters of the UMAP. There are some parameters which you can play around to either go deeper into clustering or just have a more bird's eye view. But to understand what these clusters are about, we can play around with the data mapping. And this is a universal tool and platform, the graph maker visualization capabilities. And let's say I just know what it is. So let me show you right away. And let's say take the best J gene and put it as a grouping. Yeah. So once it's, you see that the clusters that we have, they actually correspond to different J genes, which is something, well, that's not a surprise for this type of data. We can play around with the way how we visualize this data. I actually like this color scheme more. Yeah, and now we see something. What else we can do? Let's say just for fun, let's say let's use CDR3 lens as a color to see whatever to see. Yeah, well, something. I'm just showing how you can play using with the data using this exploratory tool to get sense of what we have in the data. Yeah, so this is clonotype space where we are playing around to understand what we have. But now let's go further and let's understand and let's first reproduce some of the results with the study. And the basic one is actually an easy one to reproduce is just to see. We have time points, we have day 0, then we have the product, and we have day 30. And the easiest result which we can get is just to see what's the number of T cells in general in each of these time points. Yeah, depending on the patient, depending on the responder or non-responder, etc., etc. And for that, we have a repertoire diversity analysis block, the basic tool to understand the diversity of our samples of our T-cell repertoire. Yeah, I'm selecting the data set. In this study right now, we have only one data set, so it's the only option here. I run the diversity analysis. It will, by default, calculate me observed diversity, Shannon-Wiener, CHA1, Gini index. We can add more metrics here with like Shannon-Wiener diversity or indices, well, different things. Yeah, it also applies the downsampling, the normalization to correctly calculate this diversity metrics. And let's go to the diversity block, which is not nice right now, but we can make it nice easily. So first of all, we are looking here on the observed diversity, and we can switch between what we are looking, CHA1, D50, Gini index, or whatever. Yeah, and as a primary grouping on our bar plot, we use sample. Let's change this. Let's use time point as a primary grouping. Oh, already something more interesting. And let's change the type of the visualization to, for example, box plot plus bin dots or plus jitter dots. Yeah, and I always do this. I always change the color. I don't know. I just like this color more. What we else can do, we can order the groups like this, for example, to see that we have a really high diversity in our data at the day 0, and then the diversity is obviously low in the product because we selected cells. Yeah. By the way, I'm not a biologist by training, so I may say something completely wrong, and I expect that you, Sander, and you, Dmitry, correct me. Okay. So, yeah. So here we see no diversity because of obviously the cells were filtered. And what else we can do? We can also add some statistical analysis to the visualization to see what's to bring some statistics on the plot. Yeah, but then let's go further. Let's do the following. Let's remove all those tests and let's use the responder versus non-responder as a secondary grouping to see how the diversity of the immune of the repertoire differs in different groups of patients. Let me bring ACTP before day 0, day 30. Yeah, and well, we see that in the so the first group is the complete responder, then we have the second is a non-responder, and the last one is a partial responder. And in the complete responder, that's the first group, so where the primary grouping is the time point, and the secondary grouping is the by the way, we can change the colors and do it in different way. In the day 30, the group of complete responders have the really lower diversity of the immune response, which is, I guess, something as far as I know, presented in figure 1E of the original paper, this specific results. And the diversity, the number of different T cells is goes down for the complete responders after the therapy. This is one of the instruments, repertoire diversity. There are others. We can, for example, do some standard repertoire analysis to see whether we have some biases among the groups of responders and non-responders in the gene usage. Let's say we add the VG usage block to see this, run it, and while it is calculating, should be fast actually. This gene usage is not the big deal. We see the heat map of, for example, V usage, with the clustering. Let's use the response as an annotation layer. It's one way to see this. So we see that the complete responders, they are grouped here. This is a group of responders. Or we can do other way around. We can use response as a grouping to see whether there are some patterns in the gene usage among those groups. And for example, in the group of complete responders, if we zoom in, we see, and in general, in the group of non-responders, in general, this is a general pattern. There is some gene which is definitely more presented like compared to the others. This one is, I believe, just judging by the whole picture. As far as I can see, this one is more represented in general in the group of complete responders. What we can do, for example, let's copy this gene. Let's copy this gene. Just for fun, let's go back to our clonotype space. And let's use this best V gene as a highlight. I'm copying it here to see how this gene is actually present. It's calculating. Is present in general on our space of all clonotypes. Well, we see it's pretty well presented in all clusters of T cells and in all combinations of the G genes. So nothing special about this specific gene. Fine. Let's go further. And the next basic thing that we want to see which of the clonotypes, which specific clones or clusters of clones were selected for the product, for ACTP, for the therapy. How we can do this? We can do this using the enrichment app that we have, clonotype enrichment. So this is the app allowing to identify the groups of clonotypes which are expanded in under certain conditions, and our condition is ACTP time point. And but before this, another thing which we typically do, we want to group clonotypes together, similar ones, based on their sequence similarity. And for that, I'm using the clonotype clustering app. I'm selecting the CDR3 amino acid sequence. That's the one we're going to use as a measurement. The sequence we are comparing to cluster similar T cells, and let's do blossom comparison, meaning that the protein properties of the amino acids will be taken into account when grouping them into single clusters of similar ones. And well, given this as the T cell data with CDR3 only, I'm going to set the minimal identity of two T cell clones to be joined into a cluster as 90%, and the coverage threshold, meaning that the length of the CDR3 should not be too far from each other. This will just allow me to group T cells in clusters for further more, let's say, more high-level analysis. So while I'm running this, it will take a few seconds to run. Well, it actually takes more time. The clustering of almost 1 million of T cells, it's quite large. Yeah, and here we see our clusters. For every cluster, we have the centroid sequence. We have the size of the cluster. We can actually click on the individual cluster to see what's the content of the cluster. Yeah, we see here we can see how, in general, the size of how the clustering performs. This is a histogram with the distribution of the cluster size. How many clonotypes? How many clusters of this size, and we see, well, pretty even distribution. This is something we normally expect. The cluster plot, we will see like this different clusters. And now let's use these clusters to track the enrichment and to find those clusters of T cells, which are the ones that were taken into the product. Let's select the number of reads in cluster as the abundance measure. So we are tracking the clusters now, not the individual clonotypes. And as a condition column, we're taking the time point, the column that we edit in the metadata, day 0, ACTP, day 30. And the condition order in our case is day 0, ACTP, day 30, meaning that we want to find those clusters which are well, we will see right now, which were selected for ACTP and then which should present in day 30. So it's running. It's also, well, again, it runs on 1 million, almost 1 million of clones, so it takes some time. And if we go to the frequency line plot here in the clonotype enrichment, and change the order of groups to this one, then we immediately see that certain clusters, and again, I'm doing this, and the certain clusters, yeah, they are really highly abundant in the ACTP samples. And so the clusters that were enriched in the product, and then they present in day 30. So this is something we expected. And now we are interested. We can also look on those clusters of clonotypes in the different visualizations in the clonotype enrichment block. And now we can actually take our clonotype space, move it down below the enrichment block, and use the calculated enrichment, maximum cluster enrichment, as a, for example, filter. By the way, if we go back here in the enrichment bubble plot, we see that some of the clusters have an enrichment level of almost 16. And enrichment is a log two-fold change of the abundance. So some of them are really well abundant. So let's look here on the clonotype space on those T cells which are well enriched, and we have them here. So we filtered for them and we have them here. And so this is actually the cells that were selected for the therapy. And now we are going to investigate them deeper and understand what they actually are. We can actually use a lasso tool and a multiple sequence alignment instrument. We can bring the V genes, the other many other properties here to see what's actually this sequences are about. But before that, let's use the detect tool to understand whether we can predict the specificity of whether we know whether it's possible to see what is the specificity of those T cells that we selected and marked as enriched. And for that, we have a detect block available in Platforma, which you can install. That's the really powerful tool allowing to predict the specificity of TCRs. We select the CDR3 amino acid sequence. That's the only sequences that we have in the project. It's the TCR beta sequences. There are some advanced settings that you can check, like you can put the epitopes of interest to test against them. You can also, will be available very soon to use a custom database for the prediction. And yeah, let's run it. Since I already run it in a project, in a new backup project that I have, the calculation will be almost instant. Yeah, and now let me move down the clonotype browser and clonotype space blocks. Yeah, so ImmuneWatch detect computed, so finish the calculation. And if we look in the clonotype browser on our samples, then for every clonotype, we will have additional columns generated by the detect tool. This is basically the detect antigen, which tells us what is the antigen, the predicted antigen for this specific clonotype. The score of the detection, which is very important to understand for us to understand the confidence level of this, the confidence in this prediction. Yeah, and now all this information, the antigen, the score, the epitope, and there are also other columns generated by detect. We can use them in our downstream analysis to filter, to group, to do various things. But before that, let's do the following. Let's annotate our clonotypes right here in the clonotype browser using the annotation tool to assign some labels to the clonotype that we have. Since I already know what I'm looking for in this paper, I will do this right now, but the usual way how we do this, we go to the clonotype space or some just other visualizations, and I will show some of those visualizations. We use the detect antigen, as a for example tooltip, yeah, here. And when we zoom in into our clonotypes of interest. Let me do something like this. And when we zoom in into the clonotype of interest, we actually see and can find some important information about those T cells. Like for example, we see that there is marked antigen, and actually if we scroll here, if we play around with the data, we will see that many of those clonotypes that we select here, which are supposed to be the clonotypes of interest, they have this marked antigen. And that's what I'm going to do here in the clonotype browser with the annotation. I'm going to do the annotation of my clonotypes based on the antigen. And I call this annotation antigen specific, the annotation scheme. And the first what I want to do, any antigen. I want to filter the clonotypes which have any prediction for the antigen. And let's use detect antigen. That's the column and the data generated by detect, and set it as is not NA. Yeah, it's present. So with this annotation, we select all of the clonotypes that have predicted antigen by detect. And yeah, And let's add another annotation, which we will call mark, which is the those clonotypes which have antigen. Let's me contain subsequence marked. Yeah. So we have two annotations here. We run this block. It will calculate the annotation script. Actually, that's how we called it, and assign these labels to the clonotypes based on the criteria we list here. So for every clonotype, we will have either any antigen label, meaning that there is some prediction from the TACT, or marked label, which means that this is antigen is marked, or no any label if there is no prediction.
Sorry, just for the context, the MART stands for melanoma antigen recognized by T cells. So yeah, for the context.
Super helpful. That's, you see, I'm not a biologist, so I'm not putting this under attention. So it's done. So now we have for every clonotype, we actually have this assignment, the antigen specific column, which has the value any antigen, marked, etc., etc., etc. And in the statistics, we see how much of our clonotypes in each of the samples are either specific to any antigen or specific to marked. And this information is now available in the clonotype space, for example. So in the clonotype space, here we can filter for only those clonotypes which are specific to melanoma recognized antigen. And well, you see, I applied the filter, and there are actually many. But the space is really, really huge. The space is huge, so that's why you see them like they are a lot. They are actually, if we look here, they're actually like, well, some quite a lot actually, quite a fraction. Yeah. But we remember that this is a specific study with specific patients with specific disease. So probably this is something expected. Another thing we can do, we can also use a score as a filter, let's say put 0.2, yeah, to have only those, to leave only those where we are really high confident in what they are in the prediction. And yeah, we see, well, quite less number of candidates. And to understand what those clonotypes are, we can add other information on our visualization to see whether. Oh, let's do the following. Let's take the maximum. The frequency in ACTP is a size, meaning that they will be really big if they will be bigger if they present in the ACTP, in the product, in the ones that we selected. And we see actually that some groups are here. Yeah, this is the group like with a high abundance. So those T cells, which is specific to the melanoma, were really abundant in the ACTP product. So they are enriched, and they are what actually the patient had. Yeah, and we can explore them in more details and refine our understanding of what's going on there. And at the last step, what I wanted to do here, I don't want to go deeper into this. I just want to add one more result, which is a complementary result to the study right now. And then please, let's do discussion. I want to do the following. Let's use a graph maker. That's a generic block, a generic app we have to do various visualizations. There are many of them. And let's use stack bar chart and the results from the detector to see how the total, the number of labels is huge here. The total fraction of clonotypes across samples behaves with the time points in responders and non-responders, and specifically how it behaves with the T cells specific to the melanoma. So I'm going to use the antigen-specific annotation that we generated in the clonotype browser. Well, first of all, I want to use the time point as a primary grouping, yeah, on our stacked bar plot. And as a secondary grouping, the bars, I'm going to use the antigen-specific annotation that we generated in the clonotype browser to see the proportion. This is across all of the patients, all of the samples. This is a proportion of the marked clonotypes from day 0 to day 30. And what else we can do? We can use the response as a response as a facet, for example, to see how this differs across responders and non-responders. Yeah, we can also normalize this to see this better. And yeah, for example, for yeah, well, I don't know the full biology behind this, but I believe Dmitry was.
Yeah, we see the more antigen specific clonotypes survived till the day 30 for those patients who responded well to the therapy.
Yeah, and we
so yeah, CR group have more proportion of antigen specific clonotypes for the final draw of blood for the
specifically against melanoma.
Yep.
This is one of the things that we can build here, and with this instruments, with the information provided by Detect, you can imagine how deep you can go with this to get more and more insights and results. So basically, I want to finish. I'm speaking too much. If you have any comments, Sander, regarding the biology, it will be really nice to hear. And any other comments, questions?
Yeah, thank you, Stan. No comments about the biology, but impressed with Platforma. Platform as always. I trained as a bioinformatician, and building a pipeline that does all of this would probably take a long time, and you just demo this in half an hour. So I think it's super impressive how all of these blocks actually interact with each other, and how deep, how fast you can get deep into the data. So always impressed with these demos.
Thank you very much, Sander. Yeah, well, really, since I am not a biologist and I didn't have time really to prepare well, I'm just playing around like with the data with this paper and clicking, clacking. You know, if you know really what you're looking for, then there are many things that you can focus and go into them with Platforma and with the blocks that we have. So I'm showing here only the bulk analysis, but we also have a cell ranger for single cell, for gene expression, and all of the pipeline to access the gene expression, or for example, immunoassay data. In this specific study, we know from there is immunoassay, there is assay data associated with the study, which we actually can import to the project to see which of the clonotypes are from which origin, and then use this information further to you don't see my screen, or you see my screen?
We see it.
You see my screen, yeah. So this is the assay data associated with the paper. We have a sequence, and then we have the origin of the cell. So it was done with a functional assay, and then with this assay information, we can further focus on now what are those cells that we selected here, well, for which we know the antigen, what is their origin, you know, or like CD8, whatever, etc., etc. So more and more things.
Okay, great. Thank you so much, Sander and Stan. It was a great session, incredibly engaging and informative. I really like seeing how Platforma coupled with Immune Watch Detect looks super powerful. But going off of what Sander said, what stood out to me is how user-friendly and fast it is. the ability to go from TCR sequences to meaningful insights in just half an hour is really impressive. These tools that you presented today would be useful for anyone doing immunology research, not just T cells for cancer, but other applications like antibody selection, T-cell selection across vaccine development, infectious diseases, and more. So I will drop how to access these tools for yourself in the chat, and then we can get into the questions you received. Okay, great. So we have a few questions. I think Sander maybe wants to elaborate a little bit about Immune-Watch-Detect, what it is, the algorithm behind it, because we have a few questions. Does Detect handle gamma delta or unconventional TCRs? What's the plan for extending Detect to unseen epitopes? Maybe you can just get started about the database, how it's trained, and how you have new additions in your database as well.
Yeah, sure. Thanks for these questions. Indeed, Immune-Watch-Detect actually works with both two important parts. You have a database and you have the algorithm. So we've been working very hard in the last couple of years to actually curate our own database of T cells and known epitope pairs. And I think that's a very, very important fundament to have good TCR specificity predictions. So that's one part. It's database, which is continuously expanded. We have releases each quarter. 50% currently is viral T cell epitopes, TCR epitope pairs, and the other 50% is coming from mostly oncology, but also some parasites, also some bacteria in there. Second part is the algorithm. This is the algorithm actually does database matching, but it takes it a little bit smarter than just pure exact matching. And this is also something you can actually check in a recent paper published end of last year called IMREP23 benchmark, where we actually won this a machine learning competition, showing that our algorithm is actually the best performing for doing T-cell receptor epitope predictions. So that's one thing, database, currently a lot of viral, but expanding a lot in oncology, and the algorithm is the best performative until they organize a new version of this competition. The second part of the question, Monica, was more expanding into unseen epitope predictions. This is still a very, very hard topic, as demonstrated by a new IMREP competition earlier this year, that Detect is pretty good if the epitope of interest is already in our database, and we have a few T cells known to bind this epitope. But it will be a much, much harder problem if there is not an epitope, if the epitope is not stored in the database. Although recently, I think there's still a lot of improvements that need to be done, but recently we're also seeing some performance if your epitope of interest is pretty similar to an epitope in our database. So it doesn't have to be an exact match with epitopes in database. So we're slowly expanding in the unseen epitope space, but a lot of work still needs to be done.
Okay, great. And then one other follow-up question. Can you use the same tools, Detect, and also Platforma for single cell data?
Absolutely. Both.
It's a simple question,
Yeah.
yes. I think for Detect, I can also do maybe one small extra comment there. Usually you see slightly better performance if you have an alpha and a beta chain of a T-cell receptor. Then you see a slightly better prediction scoring because you have two chains to actually make a prediction. But most of our use cases is actually on both sequencing beta chain only, so that works very, very good, but it also works out of the box for single cell.
Okay, great. And next question is for Stan. What method is used to build the UMAP plot for the bulk data?
Dimitri, it's better you answer.
Yeah, I can answer that. So we use the sequence distance as a main source. So the UMAP shows closer sequence-wise clonotypes together on this 2D plot. And UMAP algorithm is fed with K-mer-based metrics, and then it uses parametric UMAP, so it learns the embedding kind of from that high dimensional K-mer space into the less dimensional UMAP space, and then also can apply those learned embeddings to also unseen clonotypes as well.
Yeah, so basically it's a two-stage pipeline. As Dmitry said, the amino acid sequences of what is defined the clonotype. In the case of this study, only CDR3 sequence was sequenced. In case of full length data or single cell data, then it will be like the full length. Yeah, it is converted into the K-mer count vectors, and then there is a dimensionality reduction using the truncated SVD. Yeah, and then the standard UMAP is done. So basically that's it.
Great. Final question for Sander. For epitope discovery of TCRs, is there a bias towards antigen selected from the database, such as MART1, which is, for example, highly present, and as well as viral epitopes? What is the advantage compared to GLIPH2?
Yeah, sure. So yeah, because Detect is a seen epitope tool, we have to trust on the database. So if we, for example, MART1 is an antigen or an epitope, or we have multiple epitopes available with a lot of TCR sequences known to bind epitopes of MART1. So this one is actually, we have high confidence in finding T cells that map to MART1 because there's just a lot of data in the database. Same with several viral ones. I think SARS-CoV-2, EBV, we have a lot of data, and so we're pretty confident if we do a call on this with a high score, we're pretty confident to catch them. What's hard is to catch epitopes that are not in the database. So it's purely database driven, and we can get some deviation of what is in the database. So for example, we're working a lot on KRAS epitopes. Neoantigens are by definition hard because they're not in the database. So you have to figure, so there's obviously some kind of bias to what's in the database, but we're working on expanding the database to have more coverage. Then comparison to GLIF2, I think GLIF2 has been a powerful tool, but it's also quite old. I think Detect, it's just 3 years old. It's a whole new experience, I would say. So you don't have to define what antigens you want to scout for. You just run it on a whole sample, and in combination with our database, we've shown actually that it's best performance. Although GLIF2 was not taken into consideration in that competition, but I think Detect as a new, more agile tool compared to GLIF, which is of a certain age.
Thank you, Sander. Okay, so we don't have any more questions, and we're really just at the top of the hour. Is there anything else you guys would like to add, or can we wrap up this exciting session today?
I already spoke a lot.
I'm fine. I enjoyed it. Thank you.
Thank you.